Perfect. Well, I think pretty much everyone knows me here, but I'm Kit Church and I'm the Community and Research Engagement Manager for the TBRS community. And I'm really excited to welcome everyone here today to hear Dr. Sarah Waldvogel's about her work and more about Mosaicism in TBRS. Um, so thanks for joining us, Sarah. I'll introduce her to everyone who hasn't met her yet. Um, Dr. Sarah Waldvogel is an MD PhD student at Baylor College of Medicine, where she recently completed her PhD in cancer and cell biology and will finish her medical training next year. So congratulations. She holds dual undergraduate degrees in genetics and biochemistry from Clemson University and has been honored with numerous national awards for her academic excellence and research leadership. For the past 3 years, Dr. Waldvogel has been leading research funded by the NIH to explore the role of mosaicism, which is when some cells in the body have different genetic makeup than others, and Sarah will go more into that, in the context of DNMT3A, the gene that's linked to Tatten-Brown-Raman syndrome. Her work has focused on understanding how mosaic changes in this gene may affect diagnosis, development, and medical care for individuals with TBRS. In addition to her impressive scientific background, Dr. Waldvogel is deeply committed to patient-centered research. She's worked closely with TBRS families, clinicians, and researchers, and is passionate about translating complex genetic science into information that is meaningful and helpful for families like ours. Her long-term goal is to become a pediatric hematologist oncologist, researching genetic causes of cancer and bone marrow disorders. So everyone, please join me in welcoming Dr. Sarah Waldvogel.
Thank you so much, Kit, for that very kind introduction. I will share my slides now. All right, so thank you so much to everyone for being here. I'm really excited to have this opportunity to talk to you about understanding mosaicism in the context of TBRS. And we'll have a lot of time at the end for kind of discussion and question and answer session. I'm just going to first talk about some of my work during my PhD. A clinician from our lab, Ludo, is also on the call in case you have any direct clinical questions that I may not be able to answer for you at this point in my training. So I like to first offer this visual representation of a mosaic tile artwork. As you can see, this tile artwork is composed of a bunch of different pieces that are similar to each other, but different. They come together to form a cohesive whole. This is analogous to the situation in humans, where we learn in grade school that every cell in our body has the same genetic makeup, but we know that that's really not entirely true, and that over the course of a lifetime, cells acquire changes in their genetic material that result in all of them being different. And so, really, humans are all mosaics of cells with unique genetic variants. So, what do we really mean by mosaicism in a genetic context? Well, during the course of cellular life and normal DNA replication, mutations or changes in a DNA base may be introduced. And this can occur either through environmental insult or through natural processes in the cell, like DNA replication. As this occurs, the DNA with the mutation is inherited by one of the two daughter cells during cell division. And every subsequent cell that is derived from that daughter cell will also carry the mutation or the change in genetic material. Thus, the resulting organism will become what we call a mosaic, or a mixture of wild type or normal cells and cells that carry the genetic change. So it's important to understand that the timing at which we acquire a variant determines what tissues are affected. So I want you to consider this scenario in the zygote. The zygote is the first cell of the organism when a sperm and an egg come together to make the very beginnings of an embryo. So in a classic genetic disorder like TBRS, the variant or the change in the genetic material occurs either in the sperm, the egg, or the zygote. And thus, every cell in the body of the resulting person will carry that genetic change. But imagine the situation such as if the variant is acquired in one cell of the two-cell stage embryo. You can imagine that in this case, maybe 50% of the cells throughout the body would carry this genetic change. And these changes can occur progressively later and later throughout development, with the other extreme being that they can be acquired in one cell that is already restricted to become a single organ. So, for example, you might acquire a variant just in cells that are going to become the brain. And then that genetic change will be trapped in the brain. So these have different consequences for the mosaic individual's children and reproductive risk. For example, in this scenario, the person would be able to transmit the variant to their children, versus in this scenario, they would not be able to transmit the variant to their children. And you might imagine that the consequences for the person's own health might vary depending on how much of their body is affected and which specific organs are affected. This has consequences for how well we're able to detect mosaicism clinically. So I want to define a piece of jargon for you first before we get too deeply into this. I want to talk about the variant allele fraction, or the VAF. And this is a really important number that comes up a lot. So this is the percent of someone's genetic material that carries the variant. So when we sequence genetic material, we have millions and millions of pieces of DNA, and the VAF is just the percent of those pieces of DNA that have the genetic change. And so in non-mosaic TBRS, the VAF is 50% because all cells have one copy of the normal sequence and one copy of the variant sequence. In a mosaic person, the VAF will be somewhere between 0 and 50%. So this is kind of what sequencing reads look like when we get them back clinically. We have this plot, and these different colors represent the different 4 letters that make up the DNA sequence, C, A, G, and T. And these bumps represent like how much of those, what percent of that particular base in the DNA sequence is that letter. And so we may find that the variant is low at one time or in one tissue that's sequenced, and we just see this tiny little bump of the variant sequence. Now, in the clinic, these sequencing reads are often read by computers, and they're kind of messy. There's a lot of background noise, and so the computer is going to throw this away and never even tell the clinician that we found it. They're going to say this isn't real. It's really not a pipeline that's built to detect mosaicism. Conversely, we may see that the variant is so high at one time and in one tissue that we think this person just has classic TBRS, that they have the variant in every cell of their body. And we don't realize that this bump is actually a little bit less than 50%. In either case, we're not diagnosing this person as a mosaic. And we're also kind of hindered by the fact that we almost always just check blood at one time point, and there's lots of reasons for this. Partly costs, and it's usually blood is what's covered by insurance. It's more convenient. It's generally less invasive than sampling other tissues, but it does really limit us because we can imagine a scenario where what if the variant is very low in blood at one time point, or what if it's very high in blood at one time point? And so our current clinical pipelines are really not built to explore mosaicism. So we think that mosaicism that's acquired in the embryo is underdiagnosed and understudied, but has important health implications for both the mosaic individual and their potential future children. And so we sought to explore this phenomenon more deeply in TBRS. And as I'm sure most of the people in this audience are extremely intimately familiar with, TBRS is an overgrowth and intellectual disability disorder caused by loss of function DNMT3A variants. So changes in the gene sequence of DNMT3A that inhibit the function of the protein. It leads to intellectual disability, dysmorphic features, overgrowth and obesity, a range of malformations such as of the heart and brain, and we think cancer predisposition particularly to acute myeloid leukemia, a cancer of the blood. So we sought to understand whether mosaicism exists for TBRS causing DNMT3A variants and what might be the implications for diagnosis, symptoms, and family planning for these individuals. So before I joined the lab, we had characterized one DNMT3A mosaic individual who showed differential tissue distribution of the variant. So he actually had 14 children, and 4 of them inherited TBRS. However, he himself had different levels of the variant in different tissues that we were able to sample. So the variant levels were very, very high in the blood and seemed to be pretty low in all of the other tissues that we're able to sample. So we hypothesized that DNMT3A variants acquired in the embryo would show time and tissue dependent selection. And what that really means is that we thought that they might change over time throughout the course of a person's lifespan in a particular tissue. And we thought that they might be seeded at different levels across different tissues during development. So we had a few specific questions about DNMT3A variant mosaic humans. So are the variant cells evenly distributed throughout the body? How likely is the variant to be transmitted to children? And do mosaic individuals themselves have features of TBRS? And then more broadly, how common is this phenomenon in the population, and in our case, particularly the US population? So we identified 2 new mosaic individuals, so people who had never been published in the literature or characterized before. The first individual was someone who had a novel frameshift variant near the end of DNMT3A. So this is the structure of DNMT3A. The methyltransferase domain is the actual enzymatic domain that performs the function of the protein, and it's where many TBRS causing variants are located. So this variant led to his protein being cut off early, so the protein is not as long as it's supposed to be. The other the other individual that we identified had the known pathogenic P904L missense variant. So I'm going to be talking about mosaic individual number 1 first. His situation was extremely unique in that he had presented early in life with some developmental delay, but it was over 20 years ago before TBRS was a known condition. So at that time, they were not able to give him a genetic diagnosis, and his clinicians had the foresight to actually bank longitudinal DNA samples. So they had a sample from when he was about 2 years old, one from when he was a teenager, and a couple from when he was in his 20s. And so we had the first known opportunity to look at the proportion of his blood that was composed of DNMT3A variant cells over time. And we found that the variant cells actually underwent this very dramatic expansion from almost undetectable in his blood from when he was 2 years old to very high levels in his blood when he was in his 20s. So this VAF of 40% means that 80% of his blood cells actually carried the variant by that time. This expansion did not occur universally across other tissues, and it was his levels of the variant were actually quite low in almost everything else that we were able to sample. So we kind of thought about this from a basic science perspective, and we reminded ourselves about the actual role of DNMT3A in the blood. So DNMT3A is a key gatekeeper of the decision about whether blood stem cells decide to make more stem cells or whether they undergo the process of differentiation to make mature blood cells. So hematopoietic stem cells are one of the most beautiful and amazing and fantastic cell types in the body because they are required to preserve their integrity throughout a person's entire lifespan and continue to self-renew and make more stem cells, but they also must generate about 100 billion differentiated blood cells every single day for your entire life. And so every time that those stem cells divide, DNMT3A is helping them make that decision of, are they going to make another stem cell or are they going to make differentiated blood cells? So you can imagine if you lose DNMT3A, stem cells actually prefer to make more stem cells. They undergo the process of self-renewal instead of differentiation. And the differentiation that does occur is myeloid biased, which means that it makes certain types of blood cells over others. If you know a little bit about what kinds of cells are in the blood, it means that it doesn't like to make as many T cells and B cells, and it prefers to make more inflammatory, like neutrophils and monocytes, etc. Um, and so it was not entirely surprising to us that in this person with DNMT3A loss of function, that this very dramatic expansion in the blood would occur. What astonished us was the rate and the fact that these DNMT3A variant cells actually seem to overtake almost all blood production. We wondered if this fact could have actually led to an earlier diagnosis if some other methods had been used. And so we performed something called EpiSign, which is a clinically available test that uses methylation signatures to identify TBRS from a blood sample, and it can differentiate it from an array of about 70 other genetic conditions that may have similar clinical features, but have their own unique genetic, what they like to call episignatures. Since DNA methylation is a mark that's placed on DNA and is like the main role of DNMT3A. So loss of DNMT3A actually really changes this methylation pattern on DNA. So as the DNMT3A variant cell population expanded in this individual's blood, his blood acquired a TBRS methylation signature. So at time point 1, his signature matched the signatures of a bunch of healthy controls as well as his unaffected sibling. Over time, at the later time points, his blood actually changed to match a TBRS signature. And so many years before he was clinically identified, if this test had been run, potentially he could have received a diagnosis. And so as the percent of his affected blood increased, his blood became more TBRS-like. And now I'd like to sort of consider the opposite scenario. So this is the second individual that we profiled, a woman who had the P904L pathogenic missense variant. She was identified because she had two children, a brother and a sister with TBRS. And then a sample of her blood was sequenced, and she was told that she also had TBRS. She was very confused by this because she didn't have any of the features that her children did, and she didn't understand how she could have the exact same variant at apparently the same levels in her blood, but not have any of the features that had defined her children. And so we looked at the actual variant allele fraction in her blood, and we found that it was less than 50%. So 50% would represent a true case of TBRS, but hers was actually lower. The computer just hadn't flagged it because it was close enough to 50%. And we actually encouraged our clinicians to get a buccal swab, which is the cheek swab, and it was completely negative. And so that really confirmed that because she had children with TBRS, meaning that she must have the variant in her egg cells, and she also had it in the blood, but she didn't have it in the fibroblasts from the cheek. That confirmed her as a mosaic, but likely a mosaic who had undergone the same phenomenon of blood expansion of the variant cell population. So I will continue on from the human data, and I'm just going to talk a little bit about my work with the mice. And then at the end, I will give you much more of an opportunity to ask questions unless Kit would like me to pause at this point.
Yeah, if anyone has any questions, please feel free to either put them in the chat or raise your hand. But otherwise, like Dr. Waldvogel said, they'll be at the end for question and answer.
I can't see everyone. I can't see everyone on the screen right now, so Kit can stop me if anyone really wants to speak up now. Um, so as you might imagine, mosaic individuals are not, rushing to donate organs like brains and hearts and kidneys and other things that may be of interest. And it would certainly be unethical for us to sample those tissues from relatively healthy people. And so we couldn't answer all of the questions that we really wanted to in humans. And so we decided to see if we could model mosaicism in mice. And this has been a very large part of my PhD. Turned out to be quite a challenge, but we decided to see if we could establish a strategy to study mouse like mosaicism in mice. Would we see changes in the distribution of the mutant cells over time? And would we see different levels of the mutant cells in different tissues? So we established this mouse model of DNMT3A mutant mosaicism, and I'm not going to bore you with the details of a bunch of mouse genetics, but I'd like to give you just some idea of the technical aspects of this work. So this picture here is a microscope image of mouse embryos at different stages of early development, but you can see a few of them have 4 cells. So they're at about the 4-cell stage. They've undergone 2 cell divisions from that initial zygote. And we took embryos that were at the 4-cell stage and we modified just one of those cells. We introduced a red marker so that the cells would appear red under the microscope and we could find the one that we injected as well as all of the cells that it made in the resulting mouse. And we also deleted 1 copy of the 2 copies of DNMT3A in that same cell. So the red cells are the mutant cells. We then reimplanted these embryos in mice and allowed them to carry the pups to term, and they had pups with a range of DNMT3A mutant mosaicism. These injections worked pretty well, and the red marker is so robust that you can actually see it on the skin of the mice. It actually turns them pink. And when we sectioned all of the tissues of the resulting mice, we were able to see all of the cells that were derived from that original injected cell. They also are really beautifully red, and it produces some of my favorite images of my PhD.
Sarah, we do have one quick
Yes,
question.
yes, absolutely.
It's someone said it's possible that some parents were tested along with their child and the parents were told they don't have TBRS mutation, but they actually could carry it. That would mean that some people diagnosed as de novo might not be de novo, right? If that's the case, should siblings be tested?
Yes, so that is true. If a parent has really low levels of mosaicism in the blood, it might not be clinically detected. Um, the question of whether siblings should be tested. So siblings cannot inherit mosaicism. Mosaicism is not inherited. It's something that happens in the embryo or in the adult organism. And so if one of your children has TBRS and the other child doesn't seem to have any features, they seem fine, it's very unlikely that they have TBRS, I would say, and they cannot be mosaic. Like they can't have inherited mosaicism from you. So I would say that probably the sibling does not need to be tested if they don't have any clinical features of TBRS. Okay. Um, so there was really wide variability in the mosaicism that was induced in the mice. So we have mice with a range of mosaicism in their blood, all the way from almost 0 to like 80 or 90%. However, despite this range, our mosaic mice actually lacked features of TBRS. So on the left, I'm showing an image of a wild type mouse, a mouse with no mutation, and a mouse that in every cell of its body has a deletion of 1 copy of DNMT3A. So this mouse on the right is one of the mice from our TBRS model system. You can see that it's very overweight compared to the other mouse. And if you measure its bones, they're longer, and it recapitulates many of the features of human TBRS. Our mosaic mice did not become significantly overweight compared to their siblings, their uninduced siblings. And additionally, they passed all of the behavioral tests with flying colors, all of the behavioral tests that the fully heterozygous DNMT3A TBRS model mice do not perform as well as controls on. And so this was really interesting to us because it is kind of a similar thing to what we've seen in most of the human patients who have been described as mosaics, that they don't really seem to have the classic obesity overgrowth, and they have either milder neurocognitive features or they don't seem to have any clinical symptoms at all. So some of the key takeaways from this mouse model is that our mosaic mice were normal weight. They performed normally on tests of cognition and behavior, and they appeared to be pretty healthy. They seemed to do generally fine out to middle age. Uh, and one thing I'd like to highlight is that we tested whether the mosaic male mice could transmit their mutation to their offspring. And while they could transmit it, it was at the expected rate. So we were a little bit concerned that the DNMT3A variant cells would expand in the sperm stem cells in the same way they expand in blood stem cells. And that would have been concerning because that would predict that the longer a man lived, like the later he had children, the more likely he would be to pass on the variant. And at least in our mice, we don't have any evidence from humans, but at least in our mice, we did not see that occurring. What we did see in the mice was that the DNMT3A mutant cells expanded very dramatically in the blood. So in our wild type controls, where we just turned the cells red, we didn't delete DNMT3A. The cells have nothing wrong with them. They're just red. There's sort of random fluctuation across the major blood lineages. So each of these lines represents a single mouse that was sampled at multiple time points, 4 weeks apart. In our mice that also had the DNMT3A mutation, there is expansion in every individual mouse across all of the blood lineages. So that's really consistent with what we see in the humans.
I got um you
Sorry. Okay. Um, so coming back to humans and population dynamics of like how common is this DNMT3A variant mosaicism broadly. A key challenge is to distinguish this from something known as clonal hematopoiesis. So it's possible for an adult to acquire a DNMT3A variant in just one of their blood stem cells. So they didn't have it as an embryo. It's not embryonic mosaicism, but it's a pre-leukemic condition where a blood stem cell that's on its way to cancer can acquire a DNMT3A mutation as an early event. And it can expand in the bone marrow and contribute to the peripheral blood. And so this is actually pretty common in very elderly individuals. Um, but what's unique about DNMT3A variant mosaicism that was acquired in the embryo rather than an adult is that the expansion occurs at a very young age, so your 20s versus your 80s. And the expansion is very large. So even dramatic clinical clonal hematopoiesis might only reach like 10 to 20% of the blood versus what we're seeing in our mosaics is that it's more than 80% of the blood is composed of these variant cells. And so this gave us an idea of how we might be able to identify people with this condition in population databases. And so we looked at two really large US-based population databases that have the genetic information of many thousands of people. And we were able to identify DNMT3A variants in 8,000 of those people. But we set a cutoff where we looked for very, very large DNMT3A blood population, DNMT3A variant blood populations, and we looked for it in young people, so people younger than 40, where more than 50% of their blood was composed of DNMT3A variant cells. And we found 33 people, which doesn't sound like a lot of people, but if you extrapolate that to the entire US population and say, okay, well, that was about 2 in 10,000 individuals, that means like about 30,000 people under the age of 40 just in the US might be undiagnosed embryonic mosaics just for DNMT3A variants. This isn't even thinking about all the other thousands of genes in your genome that you could possibly also be a mosaic for. So we really think that this is a rare, but it's a rare disease, but rare diseases, when human populations are large, actually affect lots and lots of people. And so we think that this is a really important condition to consider diagnostically. So I just want to get a little bit into like taking this information, what does this mean for you and your family and your child? So there's a couple ways that this could play out. Perhaps you have multiple children with TBRS, suggesting that some frequency of your sperm or egg has the causative variant, and you passed it on multiple times. We contrast this to sporadic TBRS cases where the DNMT3A variant was acquired in a single sperm, a single egg, or in the zygote of the child. And that would mean that your risk of having another child with TBRS was almost zero. But in the case of mosaicism, the chance of having another child with TBRS is higher than zero, but will depend on the percent of your sperm or eggs that are affected. Another possible scenario is that your child was diagnosed with TBRS and subsequently they tested your blood. You were told that you also have TBRS, but you don't seem to have any of the symptoms or clinical features that your child does. And so that may mean that the variant is present in your sperm or eggs and it expanded in your blood, but it's present at very low levels in other organs like your brain. So I touched on this a second ago, but what does mosaicism mean if you were planning to have additional children? The likelihood of having more affected children would vary widely depending on the percent of your sperm or egg cells that were affected. And as I mentioned, in the male mice, we didn't see expansion of the variant in the sperm like we see in the blood, so that's good. In female mice, we don't really know because it's a lot harder to breed female mice enough to get these statistics on that, basically. For males, sperm could actually be tested. It's pretty easy to clinically access sperm. So you could actually test it to see the percent of it that it's affected, but this is generally done on a research basis right now rather than as a clinical test. And for females, unfortunately, eggs are really hard to sample and there aren't that many of them. And so prediction is much more difficult. A genetic counselor can offer much more detailed guidance about this topic, but if any of this applies to you, our team would be happy to provide a letter to you explaining some of the specifics of DNMT3A variant mosaicism if it would be at all helpful for your care, because particularly the blood expansion phenomenon is not really widely appreciated among physicians, and these results aren't yet published. So we found that it's a topic that's a little bit confusing and different for clinicians, and so it's not something that's always being really appreciated in these cases. And then, even if you're not planning to have any more children or any children at all, what does mosaicism mean for you specifically and your health? And I will have to say that there's a lot of unknowns here. It seems that fortunately, most mosaic individuals are asymptomatic or kind of minimally symptomatic, and our mosaic mice were healthy to about mouse middle age, which is as long as we kept them. So our speculation would be that there's a possible increased risk of blood cancer in DNMT3 variant mosaic individuals compared to the general population, because basically what you have is very large clonal hematopoiesis, and clonal hematopoiesis does predispose to eventually developing blood cancer, although certainly most individuals with clonal hematopoiesis will not go on to develop blood cancer. Potentially could be at increased risk for some of the other health conditions associated with clonal hematopoiesis, which is associated with an increased risk of cardiovascular disease. There are also some targeted therapies that are being tested for clonal hematopoiesis. Potentially, embryonic mosaic individuals could benefit from some of those same interventions to try to prevent the dramatic expansion of the variant cells in their blood. I would say that you should monitor for symptoms of leukemias and make sure that you and your doctor pursue any unusual findings more aggressively than you might otherwise. And so just to kind of wrap up, this is a visual that I like of how the effects of mosaicism probably scale with the dosage of variant cells. So you can go from being completely unaffected, where you have 0 variant cells in your body, to having TBRS, where every cell in your body is affected. And mosaics likely fall somewhere in the middle of this scale, and the scale probably differs depending on what tissue you're talking about. So it's this very complex phenomenon that we're learning more and more about, and we think that that's why mouse and cell culture models can be really valuable, because there's just simply not enough people and not enough tissues from those people to really completely understand this in the human sense. So that's one of the things that I really enjoyed about my PhD, and I'm just so appreciative to this community for your generosity and your generosity with your samples and your time and your data, and admire your resilience with the challenges that many of you all face. And yeah, it has meant a lot to me to work with all of you. So I'm happy to answer any questions during the question and answer session.
Awesome. Thank you so much, Sarah. That was like really insightful, and I think that was a really complex like topic that you explained, I think, in a really great way.